in his 1932 science fiction called The Brave New World. Aldous Huxley attempted to explore how life might be in AD 2540. A world where babies are genetically engineered to fulfill a specific role in society. Less than 100 years since its prediction, acclaimed journal Nature in 2011 declared genome editing as the method of the year. Following this, in 2015, came the breakthrough called the CRISPR-Cas system for gene editing, which is expected to revolutionize the future of medicine. This brings us to the important question: what is gene or genome editing? The nucleus of every human cell contains a long, double-stranded coiled molecule called the deoxyribonucleic acid, or the DNA. Small stretches of this DNA codes for specific proteins and controls their heredity and are known as genes. The entire genes of an organism together constitute its genetic makeup, or the genome. From the color of eyes and hair to height of a person and predisposition to many diseases, every aspect of an organism's life is decided by the genes. Today, scientists have developed a toolbox of molecules and enzymes which can be used to edit the errors in the genome of an organism, the technique known as genome editing. Genome editing refers to a special type of genetic engineering in which DNA or genes are inserted, deleted, or replaced in the genome of an organism using specifically designed enzymes. The enzymes function as precise molecular scissors. They are guided to bind to specific sequences by proteins or RNAs and create site-specific double-strand breaks at desired locations in the genome. In this way, mutated or faulty genes can be excised out of the genome. The cell repairs the breaks created in their DNA by two mechanisms called non-homologues end joining, in which enzymes directly join the DNA ends in a double-strand break, and the homology-directed repair, in which a homologous sequence is utilized as a template for regeneration of missing DNA sequence at the breakpoint. When the cell attempts to repair the breaks, new sequences or mutations can be introduced into the targeted DNA region to create new and tailored genes. The enzymes used to cut genes are known as nucleases, and presently 4 types of nucleases are used, namely meganucleases, zinc finger nucleases or ZFNs, transcription activator like effector-based nucleases or TALEN, and the CRISPR-Cas system. While various kinds of meganucleases are derived from microbes and they recognize and cleave long specific sequences in DNA that range from 12 to 40 base pairs, ZNF and TALEN are based on the use of artificial restriction enzymes. The artificial enzymes are created by fusing 2 enzyme subunits. While the first subunit can be designed to target and bind to specific desired DNA sequences, the second domain acts as the molecular scissor. The most recent technology that has taken the world by storm is the CRISPR-Cas technology. The technology makes use short repeated sequences called CRISPR to make special programmable transcription factors that allow scientists to target and activate or silence specific genes and Cas9 enzyme that can cut the genes. The potential applications of gene editing are many. While researchers plan to use gene editing to modify plant genomes and create high-yielding crops for food and fuels, the most anticipated application is the gene therapy. Researchers have already demonstrated the power of TALEN to create designer T cells of the immune system to fight cancer. Doctors at London's Great Ormond Street Hospital have successfully treated a 1-year-old baby suffering from incurable leukemia using designer T cells from which undesirable genes has been cut out. Researchers from the Kunming Biomedical International has demonstrated how CRISPR technology can be used to edit genes in fertilized primate eggs to create designer monkeys. The day is not far when gene editing technologies can be used to edit the genomes of embryos and remove diseased genes, paving way for a world without genetic disorders like hemophilia, sickle cell anemia, and many more. However, presently lot of ethical concerns surround the use of this technology, and experts fear the misuse of the same. But armed with this technology, the day is not far when humans will be able to play God.